Excellent, awesome. Yeah, thank you, Rich. Wonderful, wonderful presentation, very helpful. Yeah, so hi everyone, my name is Basheer. As Rich said, I'm in the Luca Pinello and Daniel Graver's lab. Currently a PhD student, and I've been working on designing some of our variant targeting base editing screens, especially in collaboration with some other labs in IGVF. And in this tutorial, I kind of just present some of the insights that we have learned from the design of these screens. Of course, this is ongoing work, and just kind of keep in your mind that the kind of the principles I'll introduce in this tutorial may change and improve over time. So just one thing to keep in mind as well. So first, I'll start with an overview of how these screens are conducted. Rich may have highlighted a bit of it already, but typically we use cytosine active base editors, and in review cytosine perform cytosine to thymine transition mutations, while adenine base editors perform adenine to the guanine transition mutations. These can be used to characterize variance and assess their function, hence their utility in IGVF. So generally the process for how these variants are interrogated is, first to define your variant set. So this can be by based off of GWAS or an EQTL study. And then from the GWAS, you can maybe pull the genome-wide significant need variants, potentially variants in LD proxy or fine mapped variants. And then, as I'll focus more in the rest of this slide, you then design your guides to be able to install the variant of interest. And then once you clone and prepare your guide library, you can then express these in your base editor expressing cells. And say if you are assessing variants which may affect proliferation, you can then perform a growth assay, and then sequence the guide counts and compare their abundance to be able to get a sense of which guide causes enrichment or depletion. So let's begin at the foundation of how guide design works. And this is with the design of the protospacer sequence with a compatible PAM requirement. So the protospacer sequence is typically 20 nucleotides long and is important for Cas9 binding and dictates where in the genome to target. However, the binding is also dependent on the PAM sequence, which is 3' flanking the protospacer sequence. So if the PAM requirement is not met, then no binding and thus editing will occur, such as the NGG PAM for wild type Cas9. And then the limitation of base editors. It's good to keep in mind that the limitation of base editors is that it can only install transition mutations, as well as the needing to fulfill the PAM requirement. So, for instance, this was also shown in Rich's slides, but kind of in review, about 58% of pathogenic ClinVar variants are point mutations, and among those, 47% are A to G transitions, while 14% are C to T transitions. And then if we look to see if, how much among these have a compatible PAM, around 26 to 28% of these variants do have a compatible PAM with wild type Cas9. So this is something to keep in mind in your screen when you're determining which variants you can install or not. And then also, as mentioned, to be able to increase this 26 to 28% proportion, you could potentially use a Cas9 variant with a more relaxed PAM constraint, such as SPG or SPRY Cas9 developed in Ben Kleinsteiber's lab. So now kind of segueing into guide design, some things to keep in mind is that, typically you will want your target base to be within the editing window. And then additionally, if there are other Cs or As that exist within the editing window, then it is possible to get bystander edits. And this is especially important when you want to interrogate the function of variance, is that if there are multiple edits, you want to be sure, which one of those edits attributes to the phenotypic effect that you're studying. And then also target bases may have reduced editing depending on the sequence context. So this may be another thing to keep in mind as well. So based on large reporter libraries assessing the editing of base editors across a large set of varying sequences, the editing of base editors are typically within the 4 to 8 range. Here is illustrated for both ABE as well as BE4, which is a cytosine base editor. And the numbers correspond to the normalized activity across the protospacer window. And activity is specifically the frequency of A to G or C to T transitions, depending on the editor type. And it's also important to note that these editing activity profiles can change depending on the type of editor that you're using. Especially Evo BE4, which is a more active editor, has a larger editing window, or E83AB4 has a more precise editor, so it has a smaller editing window. And not shown here, but these editing profiles can also change depending on the cell type that you're just transfecting, that you're editing in as well. If you're interested in kind of looking more into the determinants of base editing outcomes, you can check out this paper. This paper also has developed a model. I think I did see a question in the chat. They have also developed a model to be able to predict base editing efficiency as well as to predict outcome as well, called Beehive. So now getting into guide design. So essentially we want to ensure that we get editing at a particular variant site. So we can first generate guides that are tiled across the variant, where the variant site is in different positions of the editing window, so position 4 to 8. So therefore, we can generate 5 guides where the A or C base, depending on if the variant is ABE targeting or CBE targeting, varies between position 4 to 8 within the editing window. And then from these tiled guides for each variant, you can then filter based off of which among these guides are compatible based off of the PAM requirement. And then you can optionally filter based off of the activity, such as with Beehive, or Cas9 off-target predictions as well. So there are a few benefits for this tiled design strategy. So first is if you are using a PAM constrained enzyme, such as SP-Cas9, if you have a better chance of being able to generate a guide installing that variant that also fulfill the PAM requirement. Whereas, let's say you only generate the position 6 A or C guide. If that guide does not have a compatible PAM, then that variant essentially becomes untargetable. Additionally, you can ensure installation of the variant through redundancy. It is possible that when you're cloning your library that you may get dropout of certain guides in your plasmid library. So just to ensure that you're able to target your variant site, it's good to have multiple guides for redundancy in case 1 or 2 drop out from your library. And then lastly is you may potentially be able to distinguish the effect between primary and bystander sites. So for example, in the schematic, here, just imagine we have a target site that we want to install a C to T transition in, as well as a bystander site. And then we have the 5 tile guides varying by where the target site is within the editing window. So from position 4 to 8. And theoretically, since the editing window changes for each guide, you will get varying editing activity at each site for the different guides. So therefore, when you measure your phenotypic enrichment score, you may be able to perform linear modeling to be able to distinguish the effect size between the target and the bystander. Whereas if you only use one guide, you wouldn't be able to distinguish between those 2 effects. And then, as was discussed before, how we are actually able to calculate the editing activity is by using the surrogate reporter construct introduced by Rich, where it contains a gRNA spacer which is expressed, as well as a reporter sequence where you can be able to read the editing outcome and activity in paired end reads. So another consideration of guide design is if there are any endogenous variants within the cell line of study, in which a phased BCF file would be helpful and then being able to inform. So there are 2 scenarios to be to address. So first is if there are variants neighboring the target site, So for instance, in the schematic, we have, say, homozygous and heterozygous variants neighboring the target variant site. So in your tile guides, you can design guides which updates the protospacer sequence design based off the neighboring variants. So here I'm showing tile guides specific to homologous chromosome A, which contains both variants, as well as guides that are specific to homologous chromosome B, which just contains a heterozygous variant. So, so also the other scenario is if the primary variant you want to install already exists in your cell line. So this depends on the type of variants you're studying, but if your variants have a high average minor allele frequency of, let's say, 20% in the population, then potentially around 20% of your variants will already exist in your cell line. So in this right illustration, say we already have the transition variant heterozygous within the cell line. So instead of an A or C, it's a G or T. We could design guides that actually revert the variant. So in this case, we would instead not be targeting the positive orientation strand, but the negative orientation strand that contains C or A that we want to deaminate. And then also, since you're reverting the variant, you would have to interpret the effect size differently compared to designing a guide that would install a variant. Alternatively, or additionally, you can have guides that design the reference homologous chromosome as well. So I also just want to quickly introduce the specific variant installing base editing screen that I've been working on involving the labs that I'm showing in the bottom left. So specifically, we are targeting GWAS variants that are associated with erythroid growth traits. And we are attempting to install these variants using base editors in an immortalized erythroid progenitor cell line called HUDEP. So step one is currently ongoing. We're targeting GWAS variants in chromosome X. Chromosome X only, since these variants are hemizygous in a HUDEP cell line, since the cell line has an XY karyotype. And therefore, we don't have to initially deal with being able to distinguish between monoallelic and biallelic edits in a diploid context. So we plan to use this initial pilot screen to be able to optimize several aspects of our screening workflow as listed below here. And then hopefully, once we can optimize our workflow, we can then target all variants in all autosomes. On the right is a general workflow where we're using chromosome X GWAS variants, specifically genome-wide significant lead variants, LD proxy variants, as well as fine map variants. We then generate guides following the same strategy that I've introduced in the previous slides. And then currently ongoing, we're now performing the growth screen in our HUDEP cell line. So it'll be interesting to kind of get the results back and kind of see how how the guide design performs. As a conclusion, I just want to highlight some of the deliverables that our group expects to provide to enable other labs more easily design, perform, and analyze base editing variant screens. So no tool yet exists for base editor guide design to install variants. So we hope to be able to ship our own tool that performs this pretty soon, as well as a method to be able to analyze the screen results and retrieve those per variant scores that I was mentioning before that can distinguish between the target and the bystander. Additionally, we have also worked on designing A base editor and C base editor sensor libraries to be able to characterize the editing profile of a base editor in your cell line of interest in a relatively quick and streamlined manner. So this may be useful in deciding among base editors to use in your screen, informing guide design, as well as assisting with screen analysis. And in general, we hope to be able to provide best practices for variant screening, such as criteria for variant selection, further guide design strategies that are improved, the workflow, the experimental workflow, the actual screen analysis, etc. So I just thank you all for listening. I would like to acknowledge some of the groups that are involved in this work, as well as attention to a few of the members that I work on a day-to-day basis for the research. And if you happen to have any offline questions or interested in designing guides for your base editor screen, feel free to reach out to me or any of the labs above if you're interested.
Hi, everyone. Can you hear me? Yep. And do you see the slides?
Yes.
Perfect. So yeah, it's a great pleasure, to share our work. So today I want to talk about, more a downstream analysis, like, as bring up by many of the past presentations. We need to understand like the genotype and to measure like what we're actually doing at the intended sites. We need to have tools to do that. So I developed this tool. It calls CRISPR, that I believe can be really helpful for this particular task. So really briefly, like when we do genome editing, apart from screens, there are other more basic challenges. So one is like how do we measure the efficiency of our editor, and also can we check the accuracy. The second one, how do we visualize the result of genome editing activity? And then finally, how do we compare between editors? And so to do that, there are like a different strategy, but I want to bring up again this point that was raised also by Jesse. So when we do like CRISPR screen, especially for genetic perturbation-based screens, like base editing or normal Cas9, or if envision to do prime editing, I think these screens, they don't measure directly like the genetic perturbation, but instead we have an indirect measurement of the potential effect through sequencing the guide abundance in different populations. Also, we don't measure the perturbation at the potential of target sites. So if we want to estimate the effect of the perturbed element or individual variant, it's really important to visualize that. So, but again, what we do right now, we still measure the guide enrichment, or with these new screens that Rich is proposing, we have at least a surrogate for what may happen at the on-target site with these reporters. And another consideration is that if you just observe the guide, even though like you have this activity normalized base editing screen, or you have like predictive tools that may tell you like what may happen, you're still limited in the resolution of essentially pinpoint individual variants. And the reason for that, if you think about Cas9, you may have large index, or you may have bystander substitutions. So if you like just rely on guide abundance, you don't have enough resolution to validate that this variant is indeed functional. And I believe I want to take this slide just to pause 1 second and reflect on this point. So we need to observe the genotype. If we have like precision tools like prime editing, we can truly like say that we validate a variant. So with this in mind, like now I can talk about the tool, and like how can you quantify mutations. So there are like targeted sequencing is the way I would present, but there are other techniques that people have proposed before, like Sanger sequencing, or sometimes you just look at the gel just to see if the guide is active. But today I want to focus on deep sequencing data. So let's explain this. I mean, for many of you this would be familiar, but you have like your editor choice. For example, you can have CRISPR-Cas9 that will be targeted to your site of interest, and will introduce double-strand breaks, and this will result in insertional deletions. And with base editing, we learn instead that you can introduce more or less precise substitutions. So then like if you want to like read what are these mutations, you can design primers around the intended, on target site, and you can PCR amplify, and then you can sequence, with using, for example, Illumina. So now, you have a file that will contain all the reads that may contain your perturbations. So now, like with this file in hand, you need to like pick a tool, and this is one of the many tools that people have proposed. But the idea of this tool, and the tool that we developed is called CRISPR. So you start with the sequencing reads, and then you need to know like what is the potential reference, and the reference would be the wild type sequence, or if you have like multiple references, for example, you have a SNP, you can provide both of them, as I will show you later. And then you align each read to this reference, and then you want to after you align, you want to quantify if this alignment contain insertion, deletion, substitutions. And you can quantify in the entire amplicon, or you can actually quantify close to the expected site of activity. For example, for CRISPR-Cas9, should be close to the cleavage site. For the base editing, as we learn, there is some activity windows that you can use to just restrict the quantification. And this is important to mitigate potential confounders like sequencing errors. So now that you have this quantification, you can summarize and visualize the edits, and like you can say how many of my reads were modified or not. And this is just a summary of how active was your enzyme, and like how efficient it was. So this is the basic idea of the tool. And this is the analysis that you can do with a single amplicon. So you have like you amplify one site, and then you quantify. But when we started this tool, I mean, it was a long time ago with a nice collaboration with Daniel Bauer. And so since then, we have done many other extensions, like to cover several scenarios and several editors. And now the new version, CRISPR-2, is maintained by Kendall Clement, that is a postdoc in my lab. And there are a lot of things that you can do with this tool. And I don't have time today, but like you can read about these features in these two papers that we published a few years ago. So the tool that I will present is like this tool, it's called just normal CRISPR. So that is for a single amplicon, so a single site in the genome. But there are also other analyses that you can consider. You can have a batch mode, as I will show you later, if you want to compare different editors, or pool. CRISPR pool can be helpful to investigate on target plus a set of potential of targets, or you can also use WGS data. The key steps of this pipeline are, first we filter reads based on their quality. So we use the breadth score, trying to keep only the reads that have high quality. Then we do some adapter trimming. We usually suggest to use parent reads. So we need to do merging the reads, and then we finally align and visualize. So we have like two ways to use CRISPR. So one that is really like flexible. You can tune a lot of things. It's a command line version. We have also a Docker version, but we have also a very user-friendly website that is available at crispr2.pinellolab.org. And if you go here, you will see this interface. And essentially the way you can use this, you have to first select the right editor that you use. For example, you can select Cas9, CPF1, base editor, or prime editors. And then you have to specify which sequencing design you use. If it was pair-end reads, this is what we suggest. But you can also use single-end reads or interleave reads. And the user needs to do only three things. One, you need to upload your FASTQ file containing your reads after the experiment. You have to provide the amplicon or the amplicons if you have, for example, a SNP in the region of interest. And then you need to provide the guide RNA sequence. and then you're ready to go. You just drag and drop, this information, you click submit, but there are a lot of other things that you can change if you want. And, I will not explain these things today. So then, you click submit, you get an email, or if you stay on the website, you get a report. And, I want to illustrate a subset of the panel. The first one tells you like, oh, well, the reads align to your provided reference, and how many of the reads actually pass quality filter. So this is like, a first, QC, just to make sure that you provide the right reference, or, your sequencing run was successful. Then, we'll start from a really high level overview of, what happened, right? So here, essentially, you have this pie chart that tells you like how many of the reads were unmodified. That means that align to the wild type sequence, and how many reads are modified. So that means that will contain some mutations. And if you are trying to do HDR, this pie chart will distinguish between non-homologous and joining and HDR. So then we have this interactive exploratory view. So if you see below, you have like the full reference amplicon that you provided. You can scroll with the mouse, and you can see like where the substitution or insertion deletion are happening. And the way we represent these are with this black segment. This corresponds to deletions. And then if you have like multiple color, in any position, you will have substitutions. And then the insertion are shown with this little blip here. And so this will tell you that at this position there was substitution. Also, we also illustrate where is the guide RNA that you enter in input. So in this case, we expect the edits to happen here, and then there is also this activity window. So then what we do, like we try to show like the average, shape of the different events. So the dotted line here is the expected cleavage site. So these are data for Cas9. So you see that the majority of the indel are like close to the the cleavage site as we expect, and then there are some insertion and very rare substitution. So you get a sense of the average profile of this event in the entire amplicon. And then the most important plot, in my opinion, is this ALDIL plot that show you all the information in a nice summarized way. So in this ALDIL plot, the top row is the reference sequence, and then the dotted line is the expected cleavage site, or for base editing, the center of the window, and then each row here contain what are the alleles that you recover your experiment with the proportion. So and of course you can download all these figures and share to other investigators. So now, like with this, this was a point raised by Jesse. Like, how do I know that the variant, if I have like some heterogeneous result, that actually like this is the right variant, or the right element. So here I show you one example. So if you have this allelic plot, this is like data from this paper, like from then, and where we were providing this BC11A enhancer with this styling screen. So usually with count-based data, you get this kind of profile. There is some enrichment, so maybe this perturbation is effective. So you can take one perturbation and actually see what are the alleles. But now, as what I showed you before was for one experiment. But if you have sorted population, you can compare the relative abundance of the insertion, deletion, substitution at the endogenous site. And where you can learn, for example, in this case, we learned that like many of the substitutions were disrupting a critical binding site. So this is a really powerful way to investigate or validate the most promising perturbation coming from a screen. So I just wanted to show other plot that I didn't have in the main report that can be helpful for IGVF. For people trying to introduce substitution in coding region, we also create this frame shift splice site plots where you can see where is the cleavage site. This is the coding sequence. For example, this was one exon, And then you can see like how many in frame or out of frame you have, and also we report the friendship profile. And if you're trying to target splice site, we also have the proportion of insertion deletion substitution that may disrupt the splice sites if there are two splice sites. Another thing that is really helpful if you are trying to optimize your enzymes, or trying more than one, we have this batch mode. And in this batch mode, each row is one sample. And here we're looking at the same region across the different editors. So this is the untreated one, the control, you have the reference sequence, and then we have in this case Cas9 that may introduce insertion deletion, and then we have different editors. And you can see like the relative proportion of the editors. You can also take one row, and you can also look at the substitution frequency. And here you see, for example, for B4, the majority of C to T as expected. And you can also study the C to T conversion percentage if you're trying to optimize for this. So it's really helpful, if you're do experiment with base editing. Another thing that I believe is important, as Basheer pointed out, if you're trying to target SNPs, you may have cases where you have heterozygous events. So you may want to, for example, design a guide that target only one allele in particular. This is from an old paper, and you see you have the wild type allele, and then there's this other allele. So if you run this analysis without without the knowledge of this, you may get a table like this that would be confounded. But if you know that you have like a SNP there, and so at least you can actually deconvolve this information. And in the report we have like how many of the reads are modified for the wild type allele or unmodified, and the same thing for the alternative allele. And sometimes you may have this ambiguous category. For example, if you delete the SNP that allows you to distinguish between the two, is impossible to resolve. But if you actually have events that are not touching this position, you can efficiently essentially deconvolve this information. And in summary, I have some maybe 2 extra slides, but I just wanted to say you can use this tool to quantify and visualize genome editing activity from multiple editors. CRISPR-Cas9, base editing, and prime editing is an open source software. We have a really active community. People sometimes send bug reports. So if you try, just let me know if there are like problems. And also for the website, it should be streamlined. We have also a Docker image that you can download. And Rich, we are with time. Do we have like still a couple of minutes or?
We're running a few minutes ahead of schedule, so I think you have a few minutes.
Yeah, maybe I want to show like one thing that like before going to the acknowledgement, some like warnings, and these are warnings for all the tools. So in CRISPR-So2, we realize sometimes there are like problematic cases when you have the alignment. And just to motivate this, so the step one, as I mentioned before, given a reference sequence of your reads, you're trying to align them based on some scores, like how many mismatch, gap opening, and so on. The problem is like sometimes you have like multiple alignments that would be equally good. So then like if you don't know about the most likely based on your biological process, the algorithm can pick one of them randomly. And this can lead to these problems. In CRISPR-So1, this is one site where we were thinking maybe this guy was not really efficient, but then in CRISPR-So2, after we changed the alignment, now it was way better. And the reason, this is like scary, right? So the reason is that sometimes like if you have multiple alignment with the same score, would be better to prefer the one that put the insertional deletion close to the expected cleavage site, because this is the most likely explanation given the experiment that you do. So just to keep this in mind, And in CRISPR-Cas9, we fixed this. In other tools, this is not yet like fixed because they use like off-the-shelf alignments. So if you use alternative tools, be mindful. You should look by eye like these alignments, and just to make sure that some alleles are not missed by this other tool. And with this, I want to acknowledge my lab, in particular Kenneth Clement, that is working on CRISPR-Cas9. Daniel Bauer, we started this tool together, and it's a pleasure to work with him and improving over the years, and also the Zhang lab, David Liu, and Guosheng Yuan start talking for really helping, and of course IGVF. So I will stop here. If you have questions, I'm happy to answer, or we can move to the next segment.
Great, thanks, Luca. So as before, speak up now with questions. Otherwise, we'll just move forward, and you can follow up over chat.
I have a question. Can you hear me?
Yes, yes, Mike.
Luca, are there some examples of when the allele-specific analysis lets you see some downstream effects? Like, yeah. So, do you know of examples or publications where that like allele-specific information reveals some like downstream biology?
Yeah. I mean, that's a good point. Actually, in this paper, they were trying to. I don't know if you still see my screen. Yes, I think yes. So in this paper, this is like the phenotype recognizes pigmentosa. So they were trying to correct this allele, to essentially disrupt just the allele associated with this phenotype. So this can be a good example, for that. So here the goal would be like you want to maximize the disruption of the disease associated allele, but at the same time you want to minimize the disruption of the wild type allele. So this was like inside this gene, right? So this can be an example, and I think this is a paper. I'm not aware of other examples, but yeah, I think it's a good it's an important analysis. I mean, in this case we created the tool, but we didn't develop much the downstream analysis to associate this to the phenotype. But yeah, this would be one example, Mike.
Thanks.
Okay.
Luca, you could describe also, we have a recent preprint on off-target effects that are variant specific with therapeutic gene editing, where this aspect of CRISPR to identify allele specific events showed an off-target that was exquisitely allele specific. So I think there are relevant, even clinically relevant examples.
Yeah, yeah, thanks, Dan. Yeah, and this preprint that Dan was mentioned is another tool with Dan. It's called CRISPR-Me, and I would put the link in the chat if people want to take a look. Okay.
As soon as Moez gets the slide up, we're joining the CRISPR workshop today to tell you about a core method from our center called saturation genome editing. The whole team is here on the slide, and Moez Dawood who's a borrowed graduate student from Baylor College of Medicine, will tell you about the hundreds of parameters that he's optimized for this protocol. But I'm going to start with a few motivating slides as for why we're targeting coding variants and why saturating mutagenesis. So as Richard pointed out this morning, most variants linked to disease at this point are single nucleotide variants in coding genes. That's kind of where we're looking, right? So they're there. And many of these changes are missense variations, so changing the amino acid code. However, if you look at these missense variants in the clinical genetics database, ClinVar, you see that most of these variants are of uncertain significance. Many of them are rare, and there's just not enough evidence to interpret them as either pathogenic or benign. And they're kind of in this limbo space called variants of uncertain significance, or VUS, and they can't be used to guide treatment. This is a problem that's been growing over the years, as you can see with this gray line here on the plot on the right. Moez, you want to go? Okay. This is a problem in general. It's a barrier to the goals of precision medicine. And specifically, VUS are a missed opportunity, though, to provide life-saving interventions. So BRCA1 is the prime example, where if people who have a pathogenic variant have risk-reducing surgery, they can reduce their risk from 50 to 70% lifetime risk of breast cancer back down to the general population. And as you might have guessed, many of the variants or missense variants in BRCA1 and in ClinVar are variants of uncertain significance. So this is a problem across genes that are barely studied, to genes like BRCA1 and P53, where there's tens of thousands of papers in ClinVar or in CBI. So obviously, as part of the IGVF consortium, we're going to apply multiplex assays for variant effect in the interest of building variant effect maps that can be used. You can keep clicking all the way through, Moez, to create lookup tables to use as evidence for any possible single nucleotide variant on protein function or splicing that can be used for variant interpretation. Yeah, so the genes our center is currently focusing on come from a few places. First, they're genes that are definitively linked to disease or a phenotype, and they are actionable. So the ACMG73, the American College of Medical Genetics, has identified 73 genes for which incidental findings, so if you did a whole exome on somebody and found a variant in any of these genes, you would return a pathogenic variant only to the participant or patient, because a clinician could do something to ameliorate the risk of that variant would have on disease. We also take nominations from the Gregor Consortium to provide evidence for new genes and new variants that might underlie Mendelian disease in families. And another category where variant information can be very useful is from the CPIC Consortium, or pharmacogenes, where drug dosing might be changed by the activity of the variant. And together we hope to build an atlas of variant effects. So that would be a kind of our contribution to the catalog that could be used as evidence for variant interpretation. So next slide. Because we need to target genes across a wide range of functions and activities, right? So I just told you about the ACMG73. So there's genes that are, encode proteins that are part of the sarcomere. They encode DNA repair proteins. They encode, tumor suppressors. We need an assay that can be reused for many genes, because assay development is kind of the most time-consuming step of these multiplex assays for variant effect. And cell survival or cell growth is one such assay that has been used very successfully, but especially in yeast and bacteria. But here, we're going to be taking advantage of a haploid cell line called HAP1, where we only need to target one allele, which would makes editing the genome that much easier. And from different studies from different groups, at least 2,000 genes are essential in this line, including many of my favorites that are associated with hereditary breast and ovarian cancer. The labeled genes on this plot are actually on the University of Washington's hereditary breast and ovarian cancer risk panel. And between them, there's thousands of variants of uncertain significance that could be used for clinical variant interpretation. So we take advantage of this line for saturation genome editing, and Moez will tell you more about it. Moez, you can take it away.
Thanks, Leah. It's been a pleasure to work in your lab. Hopefully many fruitful collaborations will come of this and solve lots of VUS. So my job today is to kind of tell you about the gory details of saturation genome editing, everything you wanted to know, and probably a lot more that you didn't really care for, but that's my job and I'm going to do it. So first, I'm going to kind of reiterate where Leah left off. The problem here is the interpretation of single nucleotide variants, mainly because these are the most commonly found variants that are classified as variants of uncertain significance. As Leah showed you, the circle graph, majority of these variants that kind of like represent the entirety of ClinVar here are VUS. As we've sequenced over the course of the last decade, the canonical thinking was, or the classical thinking was, as we do more and more sequencing, more and more of these VUS will resolve because we'll have more contextual information and we'll have more information about benign versus pathogenic variants. But in fact, the opposite has happened in that you've had an exponential growth in the numbers of variants of uncertain significance. And this has been most prominent in the genes that are the favorite of LEA, and also on the ACMG 73, and also, you know, the most well-studied and most classically associated disease genes, such as BRCA2, BRCA1, APC, MSH6, MSH2, et cetera. And so a new approach, or an orthogonal approach at least, needs to be taken towards classifying these variants of uncertain significance and actually, attempting to bring to the clinic a level of interpretation that can actually help with patient management. The promise of precision medicine comes to a screeching halt when you're able to find the variant, but you have no idea what the variant does. And so in the NHGRI strategic vision for 2030, they laid out 10 very bold predictions. And number 7 here, the clinical relevance of all encountered genomic variants will be readily predictable, rendering the diagnostic designation of VUS obsolete. And this is what the UW-CAVA Center for Actionable Variant Analysis is going for. The idea that we can use these high throughput functional genomic assays to get after these variants of uncertain significance. And the particular technique I'm going to talk to you about today is saturation genome editing. And up front here, I want to acknowledge the team. I want to acknowledge Dr. Starita. It's been a great time, and you know, let's get to it. So the overview of SGE here, right? So I'm going to give you the broad strokes overview of SGE. I'm going to break it down into the 5 steps and give you every detail within each 5 steps. So first, we're going to start by choosing a target loci. We're going to select that loci and construct a variant library, an allelic series around that loci. Usually that loci for us is in a clinically actionable gene, such as BRCA1 shown on the right. We're then going to deliver that library of variants to HAP1 cells, which is the cell line that Leah talked about as being haploid. Further, in our current experiments, there's also the gene LIG4 knocked out in the HAP1 cells, and there's an integrated Cas9 in these HAP1 cells. Then the LIG4 knockout demotes NH non-homologous end joining as the mechanism of double-strand break repair and promotes HDR. And then the integrated Cas9 promotes higher efficiencies of editing at the targeted loci. We're then going to actually perform the functional assay here, right? So the idea is when you add this guide RNA and library, there's going to be genome editing to install every possible SNV in the library in the endogenous chromosomal loci via homology-directed repair. And the beautiful thing about SGE is that, yes, non-homologous end joining is a much more common mechanism to repair double-stranded breaks. But when you get non-homologous end joining, you're most likely going to get a frame-shifting indel. And we are targeting essential genes in the HAP1 cell line. So a frame-shifting indel in an essential gene is going to lead to cell death, and therefore self-selection of these background mutations out of the experiment, leaving us with only the HDR incorporated variants from our library of HDR templates. We're then going to take the remaining cells that survive, and we're going to harvest and sequence them. And we're going to quantify the variance representation at early time points and later time points in the context of this assay. We're then going to calculate a functional score, and then we're going to take these functional scores and we're going to map them. And you're going to get these beautiful sequence function maps. So in each row here is an exon 2, 3, 4, 5. In each column is a nucleotide base position. The letter itself represents the wild type base, so AAG. And then you can see the 3 other SNVs that were assessed in the assay. And down here in the legend, if the box is filled in, that means the SNV was non-functional. That means the SNV was integrated into the HAP1 cells. And over time, those cells that had that particular SNV died, and they were no longer found at later time points, but they were found at earlier time points. And over the course of this sequence function map, you can pull out beautiful biology and pull out many, many beautiful mechanisms that can lead to, many, many beautiful hypotheses that can lead to more mechanistic studies. And so this is the overview of SGE. And now kind of let me break it down for the 5 different phases. So first is designing the entire experiment, then cloning, particularly the guide RNA and the library, then tissue culture, sequencing, and finally bioinformatic analysis. So to be more clear, what we're designing here is around the HAP1 cell system. We're also going to design the particular guide RNAs that will make SGE work. We're going to design our entire variant library in a single tube containing hundreds and hundreds of variants for our targeted loci. And then we're going to also design all the primers that are required to do the cloning and the pre-sequencing PCRs in preparation for next generation sequencing. For the cloning itself, the guide RNA cloning is, not unique to us. It has been reiterated ad nauseum through many of these talks. But the way we clone our library of SNVs is quite fun, and I'll be showing you that. And then we're going to co-transfect the guide and library into the HAP1 cells. And our phenotypic readout here is the effect of the variant on the gestalt phenotype of cell viability over time of the assay. And so this is the particular readout that we are using as a surrogate to say the particular variant that we are assessing was non-functional or was functional. And then that non-functional or functional criteria can then be used as one component of the overall ACMG clinical criteria to designate an SNV from VUS to either the benign side or the pathogenic side. Getting ahead of myself though, first you gotta do some sequencing and some bioinformatics. In the sequencing, you take the cell culture pellets, you extract the DNA and RNA, you then do multiple rounds of PCR to prepare for next generation sequencing. Finally, in the bioinformatics, functional scoring and making that beautiful sequence function map. So starting with the design. So HAP1 cells are not your typical cell line. They were actually, and near haploid cells have been found in many tumors over the years. But the origin of HAP1 actually starts in a different cell line, KBM7, which was a near haploid human cell line that was isolated from a blood tumor and stably cultured over several months. This KBM7 contains one copy of most chromosomes except 8 and 15. And the idea was to take KBM7 and to hit it with the Yamanaka factors. And they were actually going for an induced pluripotent stem cell when they were trying to engineer KBM7. Instead, what they got was actually a fibroblast-like morphology HAP1 cell. So a common kind of misconception is that HAP1 cells are iPSC-like or stem cell-like, and they're really not. They're much more similar to fibroblast-like morphology cells. And in contrast to KBM7, HAP1 cells are adherent. And they lack a second copy of chromosome 8, although they picked up 2 copies of chromosome 19 in the process. And so again, HAP1 cells are near haploid, and they're very easy to work with relative to KBM7 cells and other haploid cells. So that's why they're the model system of choice. And so HAP1 cells, as we stated, are haploid, and showing the karyotype here on the right. This idea of being near haploid has several advantages. One, first, there's a rapid doubling time just associated with the HAP1 cells in general, much faster than in KBM7. Second, the idea of being haploid decreases the number of required edits by half because you have half the number of alleles. Further, more genes become essential in a haploid cell line that are not usually essential in a diploid cell line. So for example, the 2018 BRCA1 paper hits BRCA1 because BRCA1 is an essential gene in HAP1, whereas normally BRCA1 is not an essential gene in a diploid cell line. Further, when you abrogate that single copy of an essential gene in HAP1 cells, it will lead to HAP1 cell death, unless you can rescue it with an HDR template. And finally, the actual genotype that you assess using these haploid cell lines is a functionally homozygous genotype. Therefore, when you normally edit in diploid cell lines, you don't have the ability to control for a homozygous edit. Most likely, you're going to get a heterozygous biallelic edit. And so the idea of being haploid means you are truly only assessing that one variance effect on the overall phenotype of cell viability over time. Now, the cons to using a haploid cell line are, one, in the process of changing from KBM7 to HAP1, the cells became adherent. That causes some time difficulties in the tissue culture room, meaning that I have to sit there for many, many hours. 2, there's an actual reversion from haploid to diploid. So if you leave the cells in culture too long over time, if you stress the cells too many times too quickly, if you stress the cells just really harshly in general, the cells actually have the ability to revert to diploid. And what's really interesting is that the cell size will also double in size as it reverts to diploid. Further, there is an inference distance from the function queried and the molecular function underlying disease risk. So for example, the crux of assessing a gene by SGE in HAP1 is that it needs to be essential. However, this essentiality cell viability phenotype is not necessarily the most inline or best mechanism or best phenotype for assessing every possible gene. You know, many genes will not necessarily affect cell viability, but can certainly manifest in molecular, cellular, tissue, organismal phenotypes without necessarily compromising cellular viability. And so making sure that we're actually assessing genes where cell viability as a phenotype actually makes sense. So the actual design of the HAP1 LIG4 knockout Cas9 line, so there's a LIG4 knockout that compromises non-homologous end joining in favor of HDR. It's integrated with Cas9 along with a blast resistance cassette. These cells were kindly shared to us from the Sanger, and this Cas9 cassette is stably integrated into the genome. And what it allowed us to do was actually remove Cas9 from the plasmid itself. So this decreased the plasmid size by over 5 kilobases, which increased our transfection efficiency by multiple fold. And as Leah kind of started outlining here, the way we target genes is really particular. So first of all, it's important to know that HAP1 is very much addicted to genes involved in homology-directed repair, the Fanconi pathways, and DNA repair pathways in general. Second, like I was saying before, we need to choose genes that are essential in HAP1 cells. We judge essentiality by looking at the Gene Trap paper by Bowman et al. in Science 2015, and also other CRISPR screens done in HAP1, as well as in our own hands, we make sure that we can get these guide RNAs and we can use an orthogonal functional assay like cell titer glow at a unit level to test these guides one by one to make sure they actually kill the cells. And it's an orthogonal assay because the readout is not cell viability, it is actually ATP production. So it's using ATP production as a surrogate for a live cell. Further, the way we prioritize which genes to go after, right now the idea is to really go after genes where we have controls, especially missense SND controls on both the pathogenic and benign side. This allows us to calibrate our assay. And so when we originally set up an assay or set up a new target for SGE, we only will order a couple libraries because we want to make sure those couple libraries work all the way through, or that we don't have a dead guide, for example. And we usually target the couple libraries around those regions that have the most presence of pathogenic and benign missense SND controls. However, this is why this is such a problem, and functional assays like this are very necessary. There are less than 20 genes in the entire 20,000 genome of protein coding genes where you really have robust amounts of pathogenic and benign missense SND controls. And this is robust amounts as judged by the cutoff set by the ACMG. However, it's indicative of a bigger problem that if you just stick to genes where you have these missense SNV controls, you're not going to be able to assess many genes in general. So you kind of have to use internal controls, which I'll talk about later. Further, SGE is preferentially informative about loss of function. So for example, BRCA1 or other tumor suppressors, they should be having a loss of function mechanism overall in order for SGE phenotyping to make sense. Finally, one of the things we're learning in doing this is we want to make sure that we don't choose genes that have potential functional redundancy that could mask the effects of one of our missense SND variants. And then we also need a reasonable guide choice where and in the genes where we know the reference annotations are intact. And finally, just out of personal interest, Dr. Starita is very interested in the hereditary breast and ovarian cancer predisposition genes. And like she was saying, we recruit gene nominations from the ACMG 73 list, Gregor, as well as those are most clinically relevant. This is our current target list, and it reflects many of the values that we just talked about on the previous slide. PALB2, hereditary breast and ovarian cancer gene, also elevated to the ACMG last year. BRCA2 on the ACMG list. MRG15 and BRCA2 are both protein-protein interactors of PALB2, which is why we've prioritized them as SG candidates as well. BARD1, REF51D, BRIP1, ATM, very, very important hereditary breast and ovarian cancer genes. and both ATM and BARD1 are also under investigation in the Mendelian groups for Mendelian phenotypes, BARD1 in particular, neurodevelopmental phenotypes. SDHD is a very short gene, so it seemed like a reasonable candidate to start with in terms of whether we can apply SGE to the overall SDH family of genes, all 4 of which are on the ACMG 73. CTCF, FARS2, SFPQ, AHDC1, all under consideration right now for Mendelian phenotypes. CTCF for neurodevelopmental phenotypes, FARS2 for a phenotypic expansion of an individual who is currently hospitalized, SFPQ for neurodevelopmental phenotypes, and AHDC1 represents an interesting Mendelian gene. So what I was saying before is, less than 20 genes in the genome really have known pathogenic and benign missense controls. This goes true for many of the genes on this Mendelian list here, between FARS2 and SFPQ especially. But AHDC1, even though it's a Mendelian disease gene implicated in Shaw-Gibbs syndrome, there's actually a registry at Baylor that has collected over 300 individuals with mutations in AHDC1 where they have been phenotyped with Shaw-Gibbs syndrome. And many of those individuals possess missense variants, where right now we putatively believe that these missense variants in AHDC1 are manifesting in the Shaw-Gibbs syndrome phenotypes, but we want to use SGE to functionally confirm that. And so here I'm going to start now talking out of the design of kind of the HAP1 cell line and the gene choices into the actual reagents. So starting with the guide RNAs. So our priority ranking is with 4 points here. First, we want to make sure that the guide RNA can induce a double-strand break within the protein coding sequence. This is really important for that dichotomy of non-homologous end joining versus HDR. If you get non-homologous end joining and an indel created in an intronic region, that may or may not, probably not, kill the cell off. Therefore, we need non-homologous end joining to create an indel within the protein coding sequence. Therefore, those cells that do not faithfully represent an SNV that we introduced can be self-selected out of the assay. Second, the target needs to be permissive to a synonymous substitution in the NGG of the PAM site. So here on the top right, I'm showing this CRISPR guide RNA at the bottom and the PAM site here being this triple G. And so the second base in the NGG, we are changing from, it corresponds to the third base in this threonine. So we're changing this ACC to an ACG. That is a synonymous substitution that maintains the integrity of this threonine, but prevents recutting after our CRISPR guide has already created one double-strand break that gets repaired by HDR. And so that recutting phenomena could potentially alter the mutation, the SNV that we want from this HDR template. So to prevent that from happening, we use these PAM site edits to prevent recutting. Third and fourth are we try to minimize the off-target effects and then maximize the on-target effects. Many times these criteria are among the first criteria required for CRISPR-based assays. However, for us, the making sure the recutting doesn't happen is a much higher priority. And here at the bottom right, I'm showing another CRISPR guide where the AGG was not amenable to a synonymous PAM site edit. So instead, we introduced two synonymous substitutions that are within the protospacer region itself and near the cut position. And we find that this also is very good at preventing recutting. Now, the guide RNAs are chosen, and then the PAM sites are also chosen. And those PAM site synonymous substitution edits are also reflected in our variant libraries. So the SNV library has this functional purpose of acting as an HDR template to promote HDR repair of the Cas9-induced double-strand breaks in our HAP1 cell line. And overall, this library consists of every possible SNV, which we're showing kind of here is these rainbow SNVs on the right side, in the targeted loci of the gene of interest. So let's say your targeted loci was 100 bases. 100 bases results in 300 possible SNVs. And so those 300 SNVs would all be represented within our library. Further, we have about one variant per template, and each of these templates will eventually saturate the entire locus. So it represents every SNV. And then, the focus of the 2018 paper, as well as our current, thinking, is to really get after every SNV, because we know those missense SNVs are the most important for interpreting variants of uncertain significance. However, this programmed institution of SNVs into the library is a computational process. And then we basically make a list and send it to Twist, and they make the libraries for us. So we can also introduce every possible frame shift that we want in a particular region. So currently we have every 3 base deletion that is possible within the locus. And the only reason we don't have 1 base and 2 base deletions is because those 1 base and 2 base deletions will result in frame shifting mutations. And those will effectively act as if they underwent non-homologous end joining, because they will cause a frame shifting indel. That frame shifting indel will be present in an essential gene. The essential gene will be compromised because of the frame shift, and therefore the cell will die. And so we only use the 3 base deletions because those are only instituting non-frame shifting indels. And we want to see how the non-frame shifting indels, if they will still cause the cell to have a compromised essential gene, and therefore the cell dies, or some of them will survive. And so we currently have those in our libraries as well. Additionally, we can take known sequencing variants from ClinVar, from Nomad, from other databases or other rare disease registries, for example, and we can introduce those into the library as well. And so the one thing to keep in mind is kind of how I started this slide: the fixed synonymous SNV, the black SNV in all these plasmids, is in the NGG of the PAM site, and the functional purpose of this synonymous SNV is not just for the guide RNA, but also for the library. So 1, it decreases the chance that the Cas9 guide RNA complex will cleave the actual plasmid itself. 2, it ensures only a single round of Cas9 editing in the target region. That's what we talked about on the last slide. Further, it acts as an internal anchor for our bioinformatic alignment to make sure that when we pick up a short read with an SNV, that it is not a sequencing error, because we will also find this HDR edit, this PAM site edit in our sequencing read. And therefore we will know that this is a true positive read. And it will give us confidence that the SNV found on that short read is also truly a SNV that we introduced. So kind of describing what is in the library here, the overall concept is saturation. The overall concept is, in this entire loci, we are going after every mutation that we can possibly hit. And the SNV library, the entire length of the library is limited based on the length of the Illumina short read sequencing. So right now, each target region is only about 120 bases in length. And so the 120 bases is because we do paired end reading. And because of the short read size limits, we play it safe and we only do 120 bases. Now, at the end of phase 1 design here, we have the outputs of the guide RNA design, the library design, and primer design. And now we're going into the cloning. So the guide RNA cloning is very standard. There's a digestion of our plasmid. There's a gel purification of the plasmid. We then phosphorylate and anneal the oligos that we order. They get ligated and annealed into the backbone of this plasmid. We transform them, we maxi them, we Sanger them. The one thing to know about this plasmid is that it is PX459, which you can find readily on AdGene, but we were actually able to take out the Cas9. And so it took this plasmid, this PX459 plasmid that was almost 10 kilobases, and it cut it down to less than 5. And this has dramatically increased our transfection efficiency. And so the making of the variant libraries, this is where the cloning is really fun. And so you start with hap1 cells, you isolate the genomic DNA, and showing you the hap1 karyotype to remind you that's haploid genomic DNA. Then you do a PCR gradient. And what you're trying to pull out is the homology arm, region of interest, homology arm. So the homology arm here is used in part of the HDR template to allow the cell to use this template, find it highly homologous, and use it to actually repair the region of interest. And so in the process, and as it recognizes these homology arms, uses the HDR template for repair, repairs the region of interest by HDR. It will then integrate our SNV. But in the cloning right now from a wild type sequence, you essentially pull out a 5' homology arm, currently the wild type region of interest because it's from the genomic DNA of hap1, and then the 3' homology arm. This setup is approximately 800 bases of homology arm on each side of the library, and then 120 for the region of interest, as we said, that's dictated by the short read length. You're then going to do qPCR to add adapters on the 5' and 3' end, and this will allow, these are 15 base pair adapters that are homologous to the backbone that we're about to clone into, which is the PUC19 plasmid backbone. And so overall, what you get after a Gibson assembly, a transformation, a culture, and a Sanger is a circular plasmid with the wild type region of interest, 2 homology arms. These 2 adapters allowed for Gibson assembly to clone into this backbone. And now this circular plasmid is very close to actually the actual plasmids that we will introduce into the cells. In fact, it's only off by 2 bases, right? The wild type region of interest is just 2 bases different than really our plasmid library. So one of those bases is the actual SNV that we will assess, and the other base is the PAM site edits. And so in order to introduce those 2 bases, what we actually have to do here is a linearization PCR. So we set up 2 primers right up against the region of interest, and I'm kind of showing the sequence here. This sequence in the black box is the region of interest. And we have these 2 primers in the green box, the linearization primers. And these primers go the long way around the circular plasmid to create a linearized product where the backbone is now in the middle, flanked by the 2 homology arms from the hap1 cell line. And so now what you've essentially done with this linearization PCR is you've removed the wild type region of interest, and you've created a spot now to put in the actual library. And so this linearization qPCR occurs. There is a DPN1 digest that happens. This removes the background of unlinearized plasmid because DPN1 preferentially degrades methylated DNA. And because this circular plasmid went through a transformation, it will be methylated. And the PCR product that comes off of the linearization qPCR will not be methylated. And so you get this linearization qPCR, and the DPN1 gets rid of the input wild type background. And now you have a backbone here that we can use to integrate the library. Like I said, DPN1 preferentially degrades methylated DNA. And so the library itself, like I was describing earlier, we order from twist. So the gray region here corresponds to the region of interest. These red, purple, orange, blue correspond to the different SNVs. And then we put 2 primers on them. We put an amplification primer and we put a retrieval primer. And so the reason we do this is kind of going back to the same image on the right here. The linearization primer is very similar to the amplification primer. They're both right up against the library, but we use the amplification primers to amplify our particular library from the overall twist oligo pool. And then the reason we have these pink primers, the retrieval primers, is if we order several libraries that are consecutively spaced right next to each other, we don't want the amplification primer to hit in another library that's adjacent to the library of our interest. So first we'll do a retrieval PCR to isolate out the library of interest, and then we'll do the amplification PCR to amplify it. And overall, we then have this library of interest. This library represents every SNV and every 3 base deletion that we want in our library. And then we do a massive Gibson cloning with a high efficiency transformation, a large culture in a maxi prep, and then we check that plasmid library with next generation sequencing. So you finally end up with this plasmid library where you have each SNV represented from your target loci. And on next generation sequencing, you can see these heat maps where the beginning and the end represent the margin of the amplicons, and the middle represents that region of 120 bases where every SNV is represented. And so this is from the back half of exon 3 of SDHD. And so now at the end of phase 2, you have a guide RNA in a single tube. You have the SNV library that contains hundreds of variants in a single tube. And now you're going to take this to tissue culture. So you're going to start with thawing cells. And why am I telling you about thawing cells? Because hap1 cells, if you passage them too many times, will become diploid. So you have to be very careful about minimizing the number of passages. In fact, from day -5 all the way to day -1, we don't do any passages. We keep them all on the same plate and we just media swap. So then on day -1, you seed cells. And then on day 0, you transfect. You do a ratio of 1 to 4, or that's actually flipped. I apologize. It's 12 micrograms guide RNA to 3 micrograms library. And then you do an antibiotic selection for puromycin and blastocidin. BLAST selects for the Cas9 in the hap1 LIG4 knockout cells, and puromycin is selecting for those cells that were properly transfected with the guide RNA, because the guide RNA plasmid has a pure resistance cassette. And so then after 2 days, you remove the puromycin. At the time of the first harvest, you then remove the blast, because then you want to select for guide editing all the way up to the first harvest. Harvest split every 4 days from there. You go out to 21 days. And then the bottom line from this tissue culture is then you get a cell pellet at early time points and at later time points. And you're going to take these cell pellets for extraction of DNA and RNA. And the premise is then you'll be able to take those DNA and RNA nucleotides and sequence them, because they will represent the population of variants present at each of these days. And so I just want to emphasize one more time, in the actual tissue culture, in the actual co-transfection, when you transfect these hap1 LIG4 knockout cells, you are getting genome editing by one of 2 mechanisms, HDR or non-homologous end joining. If you get non-homologous end joining, which happens 80% of the time, even in the presence of the LIG4 knockout, you are getting the introduction of a small indel or frameshift mutation. That indel or frameshift mutation is causing a loss of function mutation in an essential gene, and therefore it is causing cell death. And therefore, even though 80-something% of our cells are getting edited by non-homologous end joining, that background is self-selecting itself out of the assay and leaving us with this beautiful HDR template. And so each of these cells is now colored by the rainbow colored variants from the plasmid library. And over time, you can assess cell survival of the variant in hap1 cells in culture. So over time, some of these variants will completely drop out because they will kill the cell that they've been incorporated into. Over time, some of these variants will only minimally drop out. And over time, some of these variants will be completely fine and grow akin to wild type levels because they're functionally normal. And so this is the logic in the tissue culture and the actual phenotypic readout of what's happening here. So at the end of the tissue culture, you have those cell harvests and you have your library, and you're going to essentially extract the DNA and RNA from the harvests and then sequence those. And you're also going to sequence your library to act as a baseline of what variants you incorporated into the cells. And so the preparation for sequencing is very straightforward. You have your 3 time points, for example, day 5, 13, 21. You extract DNA and RNA at the first time point. You just extract DNA at the latter 2 time points. The RNA sequencing is really meant to confirm orthogonally the presence of the variants. It's also meant to confirm the gene expression of the variant at the early time point, and it can lead to another functional score at day 5. And then you do 3 PCRs to prepare your DNA for the next generation sequencing reaction. So here I'm showing you kind of how it's a 5' homology arm, then SNV in the region of interest, and a 3' homology arm. But this scheme of homology arm, SNV, homology arm is present not just in the genomic DNA that we extract, but it is also present in any residual plasmid library that may be present from our transfection. Now, therefore, the first PCR we do is a selection PCR, and the 2 primers are shown in green here. One primer is very close to the region of interest. The other primer actually is outside the homology arm, where it can only be found in the genomic DNA and not in the plasmid library. So it essentially selects for PCR amplification of only genomic DNA templates and not any residual plasmid library. We use about 2 micrograms of template as input. We split these PCRs across multiple wells. We then do the PCRs in Ampere, clean them up. The last two PCRs here are very standard. You do one set of PCRs to minimize even further the size of the amplicon, as well as put on the Illumina adapters. The Illumina adapters then act as starting points for custom barcodes that we put, where you custom barcode every individual experiment that we want to sequence so we can demultiplex it later. And then you load onto the sequencer. Usually we use an XSeq or a MySeq. Finally, you have that sequencing information from your NGS experiment. Now, bioinformatics. Most of this pipeline is very classically similar to everyone else's pipelines, right? So you have amplicon sequencing of edited genomic DNA or cDNA, your demultiplex, you QC your reads, you do seq prep to do the merge paired end reads and also any adapter trimming. We do global alignment. We then take the cigar strings and we do variant counting. And then the three things that we are very interested in is the distribution of the different SNVs and what they mean, the distribution of known benign variants and pathogenic variants that can act as controls from actual clinical sequencing data in our experiments, and then finally making a sequence function map. So here I'm showing you a bimodal distribution of the different SNVs that are functionally classified in our SGE experiment. So the different SNVs here, just kind of reminding us here, you can have SNVs break down based on their presence in the non-coding region versus the coding region. In the coding region, if the one base change causes an amino acid change, that is known as non-synonymous. If that one base change does not cause an amino acid change, that is known as synonymous. And you can look down here at the bottom left, the synonymous variants are generally functional. So a functional score that is more negative is more deleterious. And as you can see, there are very few synonymous variants that are functionally deleterious. Now, there are some, but very few. And this is in line with the classic dogma that a synonymous SNV that does not change the amino acid will not actually will not really kill the cell or cause loss of function. Further, in terms of these non-synonymous SNVs, they break down as missense or nonsense. The nonsense SNVs are introducing a stop gain, so essentially causing a protein truncation. And you can see up here in the top left, the nonsense SNVs are generally non-functional. And these nonsense SNVs, because they cause a protein truncation, they will ameliorate the effect of an essential gene and cause cell death. And this juxtaposition of the nonsense SNVs versus synonymous SNVs, you can see it's almost a bimodal distribution of where they are on the x-axis. And this is what we use as internal controls within our SGE experiment to know whether our SGE experiment is working, especially in the absence of known pathogenic or benign controls from ClinVar or other clinical sequencing data. The missense variants then and the splicing variants then are of the most interest. So we talked about how the missense SNVs are most likely classified as variants of uncertain significance. And you can see here very clearly, the missense SNVs really span the entire gamut of the functional scoring range. And here, you can see another bimodal distribution that is in line with our nonsense versus synonymous distribution, where more likely these SNVs that are -2 or below are going to be the ones that are functionally compromised, and more likely the missense SNVs that are 0 and above are the ones that are functional and willing to work in the assay. And finally, when you disrupt canonical splice site mutations, they also, because this is an essential gene, will cause loss of function and can also act as positive controls within our assay. However, sometimes you do get these canonical splice site mutations that actually do not cause tremendous loss of function and do not kill off our cells and show up on the right side of this assay. And this is not artifact. This is highly reproducible, and we believe these are canonical splice site mutations that actually are tolerant to mutation. Additionally, I talked about we want to see how does our SGE data compare to actual known clinical data. And so here is the bimodal distribution from the 2018 BRCA1 paper, where all the known SNVs from ClinVar for BRCA1 were mapped to this bimodal distribution using their SGE functional score. And you can see here, the red coloring is likely pathogenic or pathogenic in ClinVar, and the blue coloring is likely benign or benign in ClinVar. There are very few mistakes in this, very few mistakes in the classification of these variants. And in fact, the AUC for this overall SGE of BRCA1 was 0.983, and the few variants that were misclassified were actually, on second review of the ClinVar data, it seemed that that clinical sequencing data was not as robust as originally thought. And so, the SGE data was highly reproducible, highly precise, highly accurate for BRCA1. And this is the sequence function map that resulted.
We're at 2:30 now. Is it possible for you to wrap up?
Absolutely. So last two things here. One of the things that we've been able to optimize over time is the actual sensitivity of the assay. So we're able to see in this tail from 11 to 14 to 17, the presence of hypomorphic variants. And these are highly reproducible over different replicates. We are tiling across our genes using saturation genome editing. Here is early data for PALB2. And one of the things that's beautiful about saturation genome editing is that it's able to replicate known genomic phenomenon, such as the escape of mRNA surveillance mechanisms in the last exon of PALB2, where nonsense SNVs are seen to escape nonsense-mediated decay. This data is actually very useful for clinical reinterpretation. And so Sean Fair in Dr. Streeter's group and Dr. Fowler's group has been able to use BRCA1, P53, P10 to actually reinterpret VUS to either benign or pathogenic. And these were actually done in real probands that were then returned to a private lab that were then able to affect clinical management. So success here, a genome browser track with different variant effects for actionable genes, variant effects translated into evidence for clinical variant interpretation, variant effect scores shared broadly, and overall fewer VUS in ClinVar and other databases. Thank you.
Okay, so I'll be talking today about the single-cell Perturb-Seq end-to-end pipeline using Sceptre. So this presentation was created by me and for Maya, but for sake of the time, I will be taking the directions. So before to start, I just want to do like an invitation slash advertisement. I think a lot of what you see today, the last weekend, was people like trying to assemble, trying to dig on these new techniques. And I think we have like this idea that we're still in the early days, a lot of this technique. So we have this IGVF CRISPR subgroup CRISPR pipelines that we are assembling a lot of people that know a little for the technical side, for the computational side, and trying to create like standards and how you can feed the community with these pipelines that can help everybody.
Lucas, you're not sharing the screen yet, just
Okay. So thank you. Yeah, thank you.
Now we can see.
Yeah, okay, thank you. So yeah, so it's an invitation. I think you can provide the links if people want to know the status and want to contribute. We have the IGVF CRISPR pipeline subgroup, and we enjoyed everybody. So it will be a short presentation, but we have this idea. We talked a lot about Perturb-Seq last week and how it looks like the tools that we have available in the pipeline. So we don't have actually end-to-end pipelines to solve this problem. You have like so many types of these techniques that we need to try to solve this problem, seeing these different variants in these techniques and trying to find how you can assemble pipelines. So today I'm going to show like one of the possible data sets that we can use to start to doing creating pipelines. So this paper is a 2019 for the Gasparini, for the Jay Chandri lab, and they create this K562 library when they have enhancers and they target this in this pilot screen around 1,000 enhancers and trying to see the connection with which genes are being regulated for these enhancers. So they have two guide RNAs. They have this Cas9 CRAP system, and they are checking the neighbor genes to check which genes will be affected for the guide presence. So I think it's a public, is a lot of tools already using this as a benchmark would be, and then we have like this end-to-end pipeline that we're going to show today that try to analyze this and provide results similar to the final paper. So just showing how this typical experiment would work. You have the system, then you have the lentiviral gRNA library. You have the CRAB9 system, and in this CRISPR, they use the crop-seq. I think Jesse have talked discourse about this last week, and crop-seq is one of the system where we have like something as direct guide capture that looks less sensitive to different cell types. So this is the way that they solve this problem in this paper. But we have like people already trying to develop new techniques and try to maximize the result of this technique. So briefly, you're gonna go for barcode step, get the samples, have the droplets, the barcoding, and then for this Gasparian data set, they have one part of this library being sequenced for the single cell RNA-seq. Yeah, another part will be from the library will be used to sequence the guides. So you have these two readouts that basically in the final product, you want to align this FASTQ reads for guides and transcriptome, and you would be like this product, you'd have this product that cell is like these two matrices are going to be cell by gene, and another one that's going to be cell by guide. So the same cell, since this is a high MOI experiment, can have these multiple infections, and then you would find a different number of guides in those cells. And in the end, you need to get this balance where you get a specific guide and check which genes are being affected by the depletion of a given enhancer. So in the end, you have the task of this differential perturbation quantification. Then we can use different tools to try to solve. So this experiment here, they're using this structure that is a 10x chromium reagents, and you have like a specific way that the reads will be assembled for you to detect your guides, your transcript. And that's very interesting because it's not a lot of Perturb-Seq around. So people want to start to do this experiment, and then they do the protocol and they trying to analyze this. In the very early steps, you could already detect that it's a problem or not. And that was a case that we're having recently in your group, that people have been doing like the experiment. If you check like this bottom right graph here, if you read the structure of your read here, you expect it, for example, has this poly A tail in one of your amplicons. And if you go in the manual of the Chromium 10x, you will see that you expected to see this huge variation when you see R1 here in this read for the guide. And in the very early step, you already can, if you do a FastQC, and if you get in the manual of the chemistry that you're using, could be a very nice way to troubleshooting if you are going for the right direction. So this is an example that this bottom one is a fail. The top one is when you see the poly A, you see all my amplicon process working. Let's continue with the pipeline. So how would it look like this pipeline? In this case, I will have like reads for I think I have a reversion here, sorry, but it's like this one first here should be a guide, and the other one should be the single cell. But basically, you're gonna align these two groups of FASTQ files, one against the guide reference. This guide reference will be created using the sequence of your guides, and then you're trying to match which reads are representing that guide. That's gonna be using Callisto and Boost tools, and then you're gonna have some pre-processing generate the guide table. The another one is doing just a transcriptome, like almost everybody's already used to do. You have a RNA-seq, and you need to generate a transcriptome reference. You're gonna align that. You're gonna count, and then you have for each cell, given the barcode of those cells, you're gonna have a reference in this blue table for the guide, the another one for the transcriptome. So for you got in this final, yeah, after you got this like this count, you have this task that you need to have some quality controls, and that's another moment that's important. The community is still like trying to understand what's the best way to filter this data, what's the variation when you trying to different techniques, if it's a Perturb-Seq, if it's like a different CRAP system. So how you accumulate this standards and use to have like pipelines that can be useful. So you're gonna get this tables, account table. You're gonna use that data in ScanPy to remove things that could be contaminations or poor quality cells. The final idea is create this tables that we can put in the next step that they're going to quantify your differential perturbation. So here's just some example of quality controls that we can see you can use in your count table. For example, mitochondrial reads are very important. They can signalize that you have damaged cells or dying cells. And if you have too many mitochondrial content here, maybe it's better remove those cells. So there's another threshold that different papers are using. That's like how many UMIs you need for you consider that cell as like being detected, and how many, and they have numbers here, but how I'm saying this is still, you're trying to check how it's going to change for technique, but we already have some numbers that have been replicated, and you try to use the same ones. So if you find guides on the cells, how many UMI you should find to detect that cell having a guide or not. So they have some numbers that people have been showing that are useful. And like saturation, this paper actually is using this threshold that they say they need to sequence until 20% of the transcriptomes to start to detect well the perturbations. So there's several quality control things that we can do, but that's like one of those that are most used. So I think this part is very interesting because it's why you should use covariates to analyze this kind of data. So I think last week the Weissman group showing that how the batch effect is huge on the data, how they solve that by trying to normalize that. But we still can use this covariate to help us to mitigate these different effects that we find in these experiments, in these assays. So a percentage of mitochondrial you can use like the number of detect genes that can analyze the dropouts, the total genes per cell, the guides per cell. That's a very beautiful example. I think in the end here they're showing it was a thing that we faced. When you have a cell that has a lot of guides that we expected, so you should to see the gene that's being controlled for that enhanced decreasing. The cells has, I don't know, 100 UMI for a given guide, but you see the expression of the genes going up. And then when you see that's effect that cells that has more guide RNAs, they have as well a higher amount of UMI, could be the quality of the sequence. So it's very important to use covariate, otherwise you can see unexpected effects. So okay, you have count tables now for the guides for RNA-seq, and we want to show how we could find this differential perturbation. The way that in general these tools are doing is they select those cells that has like a specific guide, a presence of a specific guide in those cells that doesn't have that specific guide. And they are not using a control cells, but they're using the another guide's presence as the control group. And the question is the presence of this specific guide changing impairing the perturbation of the genes? And then different tools have like a different ways to show this. You have this SC-Magic, that's a tool they are using, this linear transformation. It's a nice tool, it's easy to use, but it can be slow, and they're not like take advantage of the covariates. And you have this effect that we call p-value inflation. They're not treating this over-dispersion that we know that's like a problem in biological, especially like a transcriptome data. So if you think about this, you have Monaco 2ED6 was the originally using the Gasparini paper, and they have like this very nice figure for the Michael showing that biological data have this dispersion problem. And here, I'm just showing how if you have low counts, you have like a more dispersion than high amount when you have like a mean count of your genes higher. So another way to try to find these perturbations using these negative binomial models, trying to model this as a unique thing, putting the how deep the cells will sequence, the cover rates, the expected dispersion, create this model. And Gasparin show that it's a good way to use this technique, but we still have this problem that I call that this Sceptre paper is calling p-value inflation, and they say that if they get the control genes guides, they are supposed to not target any region in the genome, you still see those guides being affecting genes in cis. That should not happen. So they come this tool that we call Sceptre. Sceptre will try to do like two different things using this conditional resampling that's taking consideration the cover rates. The second one doing some adjustment in the way they treat the library depth as a cover rate. It's still like is low, but when you when you see like the results, we're not having more of that inflation in the p-values effect. For now, is the tool that we have been considering as the most trustable in terms of the results that we expected in the literature. So he's just a way. Yeah, I was talking about positive controls. He's how when you think about this over dispersion in the inflation, how Sceptre is doing well. So this Sceptre is not a tool for Pinello lab, but we're trying to integrate this in a bigger pipeline. He is like the differential perturbation, how it looks like. So these tools can be used for in-cis perturbations, in trans-perturbation. That's another field that we can discuss as well, because have like room for improvement. These tools need to be faster for calculating like the whole genome inference. So yeah, another way to, when you start to design these experiments, is try to check if the papers provide some idea of power calculation, where you have idea how many guides you can introduce, what's the best trade-off between the amount of guides that you want to screen and the target regions, and how are you losing your power to calculate the perturbations. So there's some limitations in these techniques. The tools are in the early days. The Perturb-Seq is still like an expensive technique, so you need to have this know about this trade-off and know that still expensive computation with to analyze this. So we are providing a tutorial that you guys can click this in the link, and you're trying to show this collab. It's not possible to run everything in the collab here, but if you download this for your local computer, you can try to run step by step, download the original data, and getting to the last step that we're going to use the Sceptre to calculate this perturbation. So thank you for Pinello Lab and the Power Lab, for the IGVF as well. And yeah, that's that one.